Okay, where I welcome you all to today's presentation or today's discussion on the neuron, which is actually the structure and functional unit of the nervous system. So you can see from here, this is one picture of the neuron. Interestingly, that is also another neuron. Again, this is a neuron, that is a neuron. These are the neurons. So how many, you can see there's so many variants of neurons in the shape, structure, and arrangement out there. Right. So we are going to discuss about these neurons in the heading that the neuron is the structural and functional unit of the nervous system. And let me give my details. Dr. Shakil Shams, Associate Professor in charge at the Department of Anatomy, Dhaka Medical College. Today, actually, we are going to understand what is or be able to the participants define neuron, describe the structural details of the neuron, classify neuron, and relate the functional aspects of the neurons with their structures, right? So we all know that there is a structure-function relationship, and that we'll try to find out at the later part of the discussion. So what is neuron? So as to say, as I've already mentioned, neuron is the structural and functional unit of the nervous system, which is responsible for reception, integration, and onward transmission of coded impulse. This is the complete definition of neuron. But if we just say this, it's near the structural and functional unit of the nervous system, that will do. But let us add some more points to make it more specific, that it is responsible for what? It receives the information, it integrates with the previous information, and finally, it makes an onward propagation of this information in the form of coded impulse. Like, okay. Now, let's discuss about the structure of the neuron. I'm taking the help of the book Snell's Neuroanatomy for this purpose. You see, the neuron has got a very nice and a very defined structure, though they are of very different categories in the shape and look that I have just shown you. But altogether, neuron has got in general, it has got a soma or body and some neurites or processes. These processes are called dendrites, as you can see, and this one is called the axon. So why they are called dendrites and why they are called axons? What are the differences between them? Just in short, we can see, if you look at this picture, you can see this is tapering, short, and branching. The dendrites are multiple in number. When present, they are multiple in number. So dendrites are those processes of the neurons which are short, branching, and tapering, and maybe in multiple numbers, present in multiple numbers. Whereas you can easily identify that axon is a long, where it is present, maybe long, but it is long. But the difference is that it's non-tapering, cylindrical throughout the length it is maintaining the same diameter and may be branched at the end, but not in the way. And if present there only one axon, maybe there would be no neuron having some axons having no axon, but if present it is only one. And functionally, if we consider, the dendrites actually receives the information and takes it to the soma. That's why they are called afferent component of the neuron. And the command, which is that I have told you, reception, integration, and onward transmission. So they're receiving all the information here. They're receiving the information here through these axons. Then there is integration with previous memories and previous commands and whatever. And then finally, a coded impulse goes downwards. How it is formed, in which form it is formed, I'll be telling it a bit later. But just for now on, we make it clear that it is going downwards through the axons to reach its target organ. Maybe it's a nerve, maybe it's a gland, or mostly it's a muscle, be it cardiac muscle, be it skeletal muscle, or smooth muscle. Actually, the neuron's target is, nervous system's target is either the smooth muscles, cardiac muscles, and skeletal muscles, or the glands. To be more precise, the exocrine portion of the glands. And you all know that there is some somatic nervous system which is supplying or giving target, making the target only the skeletal muscles, neuromuscular junctions. And the autonomic axons actually goes to the smooth muscles, cardiac muscles, and glands. Okay, so this is how we can classify the, in short, and with the functionality that I want to mention. With the functionality, we have mentioned the differences between the dendrites and the axon. That are the altogether neurites of the neuron. And now let us concentrate our focus to the soma. As you can see, in case of other cells, there are nuclei, the cell membrane, and also some other structures like we'll be seeing in the detail in this next picture here. Sorry. Extremely sorry. You have seen this picture, right? So structure of the neuron, we can see a very famous picture which denotes all the structures in details. In this book, it is there. And we just getting to that. But by that time, we can say that neuron has got a cell body, which is consisting of a nucleus, and surrounding the nucleus, there are some cytoplasm. And in the cytoplasm, there are some structures hanging. Those are, yes, this one is actually extremely sorry that I couldn't find it out easily. So you can see, this is the nucleus, and surrounding the nucleus, this is the mitochondria, and some deep structures. These are the nuclear substance, a very specific structure to the neurons. And there's some Golgi bodies, ribosomes, and all the structures are similar to that of the other cells. But what is the difference? And what is the specialty? First of all, we can see there these bodies or nuclear substance are actually nothing but the stacked cisterns of the rough endoplasmic reticulum. So we can understand these are actually involved with formation of proteins, because we know rough endoplasmic reticulum actually is involved with the formation of proteins. So we can predict that the neurons will be producing very huge amount of proteins. And as the protein formation or any formation structure is a very energy requiring process, then we'll need to have plenty of mitochondria, and that is there. These are there in plenty of numbers. You can see all through. But you see, there is no such dense bodies or structures, which is called axon hillock, where from the axon starts. So we can see it is free of all these dense bodies like the nuclear substances. Interestingly, another thing which is different from other cells is that they do have centrioles. A pair of centrioles are present in neurons, but they don't take part in mature cells, I'm telling. They don't take part in formation of the mitotic spindles and thereby giving rise to two cells, that is cell division. No, they don't do that. They just maintain the cytoskeletal structure. That's all. But maybe in immature, maybe not. It is true that in the immature cells, they take part in the formation of cellular divisions to a certain extent, but not in the mature cell at all. They don't take part in cellular division. And you know, in the last class, I've told you what is the consequence if the neurons divide. We'll be having fleeting memories, right? Now, let us concentrate on the Golgi apparatus function. Actually, we can show these things in very highlighted form. I have just highlighted some of the points. Let's see what are those points we need to remember. So initial substance, I've already told they are not present in except the axon hillock. endoplasmic reticulum on top of another, that is stacked on one top of the another. Basophilic, you don't understand it is basophilic. It should be basophilic. But more important is this function that they form protein. And these proteins are actually the neurotransmitters. So we can understand the currency, the currency for Bangladesh is taka, elsewhere it is dollar is international currency. So the currency of the neurons is how the communication method through which they communicate. One neuron communicates with another. That is more appropriate example is the proteins that we call neurotransmitters. So actually, the neurotransmitters are formed in the cell body. Then they are transported along with the axon to reach the next neuron by a process that is called the synapse. But we'll go into that. There are so many delicate things and detailed arrangement and detailed process of passing of one neuron's coded impulse to the next by a specific manner. We'll be discussing in the next classes. But at this moment, we just stop here saying that the nasal substance forms proteins, which are actually neurotransmitters, which goes to the next neuron and act thereby. In one interesting thing, it is called chromatolysis. Chromatolysis is a situation where as if the nasal substance has disappeared. But no, it's not like that. It is due to some fatigue or neural damage. These nasal substance goes to the periphery. Instead of circulating everywhere, it goes to the periphery, and the central part looks like as if that is clear. Lysis, the nasal substance is lysed. That's why the age is called chromatolysis, but actually it's not like that. If the injury or fatigue is removed or it can be managed, then they reorganize and this neuron gets to its functioning again. Now, already I've told about the Golgi apparatus. As in other cells, the Golgi apparatus temporarily stores and packages and adds protein to the glycoproteins. Finally, also what is important that it is synthesis of the cell membranes, particularly that of the formation of the synaptic vesicles. We must remember that they take part in the formation of synaptic vesicles. Right. So all other functions are almost similar to other cells, but this is one important thing. Remember that they take part in the formation of the synaptic vesicles. Okay. I'm just giving the summary views or summary points that we need to remember. Mitochondria is elsewhere, similar function, the cellular powerhouse, we know it. And there is mechanism, I don't need to elaborate it, I think. Now come to three things, neurofibrils, neurofilaments, and microtubules and microfilaments. So what are these things? Actually, these are the structural and other functional structure, like the neurofibrils. They are present numerous and all parallel to each other throughout the cell body, and they enter into the neurites. That means they go to the dendrites, they go to the axons, but in parallel manner. And with these can be resolved into bundles of neurofilaments. So neurofibrils can be dissolved into filaments, smaller, even smaller. And 10 nanometer, 10 nanometer structures are called intermediate filaments. And by these intermediate filaments, we can actually identify from which origin these tissue is from. Right. So neurofilaments are for keratin, vimentin, we know other neurofilaments, which are all 10 nanometers, and these are called 10 nanofibers or intermediate filaments. They are not as small as microfilaments, which is 3 to 5 nanometer, or as thick as microtubules, which is 25 nanometer. As they're in between these 3 to 5 and 25, that means between the microtubules and microfilaments, these are called neurofilaments. And these neurofilaments are other name of this is 10 nanofibers or intermediate filament, right? And by the identification, whenever the tissue identification or the source of the structure is not that much understandable or identifiable, then we do routinely, it is now even in Bangladesh, it is routinely done to identify the 10 nanofibers. So if we consider that it's a nervous tissue origin tumor or cancer, we will do the neurofilament study, and that will give us the idea. Okay. So apart from that from the clinical diagnostic importance, it is the main component of the cytoskeleton. Okay. It is main component of the cytoskeleton, the neurofilaments. Now come to a bit thinner structure. This is called microfilaments, and they are formed by actin, and they remain just beneath the plasma membrane. Okay, and with the help of the microtubules, they actually form the new cell processes and retraction of the cell processes. Okay, so cell processes goes out and retracts. That is by the help of the microfilaments with the microtubules. And now the thickest or the widest diameter of these is the microtubule, which is actually present throughout the cell body and its processes. And it is actually maintaining the cellular structure and forming the processes, as I've already mentioned. Now, what is the role of these processes and importance? There are two dynein mechanisms. One is as I've already told you, microtubules and microfilaments provide a stationary effect that permits specific organs to move by molecular motors. Molecular motors are there, two kinds of motors. One is rapid, one is slow. One is rapid, one is slow. And we know there are two kinds of transport. We can, from this picture, the previous picture, we can guess that there can be two kinds of movement, right? There can be two kinds of movement. One movement is actually from the cell body to away from the cell body and towards the cell body. If it is away from the cell body, we call it anterograde propagation. And if it is backwards towards the cell, it is called retrograde. Okay, retrograde propagation. Now, which motor is, which is a bit delicate, a bit detailed one I want to mention. That means these motors are of two kinds, as I've already mentioned. These motors are of two kinds. One is for rapid transport, other is for anterograde kinesin, and dynein for retrograde movement. But slow one is only for the anterograde propagation. Okay, now let us come to the lysosome. And there are three kinds of lysosome as in other cells. One is primary, one is secondary, and one is residual bodies. And I don't need to mention it. But what I have mentioned properly and with very much stress, that centrioles, which are small paired structures, and I've already told it is present in immature dividing nerve cells, and they are associated with formation of the spindle during cell division and the formation of microtubules. Actually, this is related to the neuroblast or immature. We all know, actually, not only you, we all know, sorry to mention that, we all know that blast cells are less mature or immature than the site cells. So the neurons are already mature one, more differentiated, so as to say, more differentiated than those less differentiated blast cells. And these blast cells are immature cells. We know the process of differentiation, right? Differentiation is a process by which one cell attains further maturity, both in terms of structure and function. And it's an irreversible process. That means when a neuroblast converts to neuron, actually, it is going through an irreversible process and making it more structurally and functionally specific. So at this stage, maybe these centrioles be helping, but when the cell is matured enough, then they only for the maintenance of the microtubules. With this short description, and plasma membrane is all the same, so I need not to elaborate any further. Let us go to the next point, that is, what is the classification of neuron? We can classify neuron according to polarity, right? According to polarity, neurons can be classified into mostly the unipolar, the bipolar, and multipolar. And as the name implies, unipolar means there will be one pole, bipolar means with two poles, and multipolar more than two poles, right? But there are another one that is pseudo-unipolar. Actually, this one is, though it is written as unipolar, it is actually pseudo-unipolar because it has got a peripheral process which takes the sensation or information at the receptor located here. It takes the information, sorry, from here actually, and then it goes to the cell body. It may go to the cell body or it may bypass and then go to the effector part. So this is called actually pseudo-unipolar. That means these two, there are two poles from two sides of this. This is a true bipolar where this is the cell body and the exons or the processes are long processes are actually formed or emerging from two specific poles. But when, just consider this one, this pole, just consider that this pole is coming to this side and that is also coming to this side. So what will happen? It will look like that, right? So it is pseudo-unipolar, we call it. And it is located in the dorsal root ganglion of the spinal nerve. This is a very famous example. And the bipolar neurons are located in all the spatial senses, like the olfactory bipolar cells, retinal bipolar cells, and in the hearing system. They are also bipolar. So they are all with the spatial senses. And multipolar neurons are the most of the neurons of the brain and spinal cord are of multipolar neuron, whereas one is axon and there are multiples of dendrites, right? And what is the example of true unipolar then? It is said that neuroblasts are unipolar in the true sense because they give rise to, let me draw one neuroblast like this. Actually, I'm not familiar with drawing with these, but somewhat is like that. Sorry. In the mouse, it's very difficult to draw. Just have an idea. So this is unipolar, right? One pole, and that is only one. So this is present in the neuroblasts and mesencephalic neuron of the trigeminal nerve. Actually, these are the examples of the true unipolar. And this one is actually for the pseudo-unipolar, which is located, once again, I'm telling, it is the dorsal root ganglion of the spinal nerves. And these are present in the spatial senses, and these are the most common neurons. According to the length of the axon, now we can see that length of the axons in some neurons is small, short, and in some neurons it is very long. Even it can be as long as 1 meter or more. Even 1 meter or more an axon can be. Can you imagine that? Or do you approve? Or do you agree with me that the axon can be even 1 meter or long? Definitely. It can be. Okay. So these long axons are called Golgi type 1, and these having short axons are called Golgi type 2. And most of the Golgi type 2 are inhibitory in nature, but Golgi type 1 can be excitatory or can be inhibitory, like these pyramidal cells present in the cerebral cortex or the lower motor neurons present in the spinal cord or elsewhere are excitatory. Whereas the Purkinje cell, with this Purkinje cell, and these Purkinje cells are present in the cerebral cortex, and they are actually inhibitory in nature. So it doesn't necessarily mean that all the Golgi type 1 cells or neurons are excitatory. Rather, some of them are excitatory and some are inhibitory. But the Golgi type 2 are mostly inhibitory in nature. Okay, now let's come to according to the shape. Again, we are saying, or we're showing the picture. You see, it is pyramid in shape. That's why it is pyramidal. It is fusiform. That's why, it's fusiform. It is stellate. This is granule shaped. So these are the shapes that determine the name or the cells according to the shape. Again, you can see they not only is a Purkinje cell in full development interneuron cells. This is an interneuron cells, very short neuron. This is called interneuron cells. Sensory neurons having the receptor, then the axon, then the cell body, and then the central process. This is the peripheral process, as I've shown you here. This is the central, the receptor here, located receptor. then it is taking the information to this neuron cell body, or it can bypass, then it goes to the through the central process to the central nervous system. That is one here been shown. Okay, so according to shape, we understand. Now, already we have discussed this point, relating the functional aspects of the neurons with the structure. Definitely you can now have got some ideas which can help you or help us to relate the functionality of the neurons with the structure. Say, for example, if a neuron doesn't have a long axon like this, so it will not be able to carry an information to a distant place, right? Like this one, it is actually located in our brain, and it goes all the way down to our spinal cord, at least, pyramidal cells. So it's a long way. So it has to have some long axons. But in case of inhibitory neurons, it doesn't need, these cells that don't need to have long processes to act. They just inhibit the neighboring structures, like we'll find in the huge amount of collection of inhibitory neurons in the cerebellar cortex. We'll find whenever we study, it would be very much interesting to find that the inhibitory neurons are usually packed or collected in the cerebellum. And most of them are small, like granule cells, like the Golgi cells. All the cells are almost very short, but they are all inhibitory, including that of the granule cells are also, is excitatory, but other than those, every cell is inhibitory in the cerebellum. So you can understand this is one sort of functional relationship with the structure, that if a neuron has to work distally, far away from its location, where from the cell body is located. Think about the spinal nerves, the radial nerves, ulnar nerves. These are nothing but the axons of the neurons whose cell body is located in the anterior horn cells of the spinal cord. You know, the spinal cord has anterior horn, posterior horn, and anterior horn is related to the motor root, which from the anterior horn that comes out, and this sensory information is entering into the posterior horn. So the anterior horn actually contains the cell bodies of the neurons whose axon will come out as the motor root of the spinal nerve. Okay, so these motor root of the spinal nerve will carry the axons, not the cell bodies. Just remember one thing very clearly, that the radial nerve, ulnar nerve, femoral nerve, and all the named nerves has got only the axons. When I cut, we are cutting actually the axon, not the cell bodies. Cell bodies are located elsewhere. They are located in the spinal cord's anterior cells. So with these, we'd like to conclude today's discussion. And I would like to thank you very much. Super thank you, should I rather say, for being with me. Thank you. If you have any queries, please raise your hand. I may try to answer. Or even after the class, if you want to ask something we need to elaborate any further, you can write in the comment box or in the questions I have formed. I'll try to solve and give the answer to that. Thank you. Thank you very much. With this, we want to end today's session. Thank you.